Hi, everybody. My name is Adam Floyd. I'm the Commercial Operations Manager for Think20 Laboratories. I'm going to be giving you a short presentation today on analyzing terpenes and hemp. So I've got about 11 years of experience in the cannabis industry. I've done a tremendous amount of research on terpenes, and it's a very large source of fascination for me. Think20 Laboratories, we are a commercial cannabis and hemp testing lab. So we have a DEA license for hemp testing, and we take in samples from all over the US. We have two laboratories, one in Irvine, California, and one in Columbia, Maryland. So to begin with, I'm going to talk a little bit about sort of how hemp is defined. So there's state-by-state regulations. Certain states set a limit at 0.3% delta-9 THC. And then there's a federal limit, which is a total THC concentration of less than 0.3%. So this is a combination of the acidic form, so THCA, and delta-9. Now, in general, there's two different major categories of hemp. So there's industrial hemp, which is used for oil, fiber, hempcrete. There's tons and tons of paper, lots and lots of industrial uses for this product. And then the other big category is CBD hemp, which is mostly what I'll be talking about today, is that as terpenes relate more closely to this. There's also a lot of interest that's growing in CBN hemp and CBG hemp, but we won't really discuss those too much today. So first, before we get into the, how we analyze these compounds, I'm going to talk a little bit about what terpenes are and how they relate to the hemp and cannabis experience. So whether you know it or not, you've probably interacted with terpenes a tremendous amount in your life. Things like pinene are found in cleaners. This is a compound that's present in pine trees and give it that very distinct piney scent. Mint, tomatoes, any sort of effervescent plant most likely contains terpenes, and they are nearly omnipresent in the plant world. They serve a number of purposes metabolically. So, they signal, they can work as a pest deterrent and as a chemical signal. So they're aromatic oils. Certain plants have only a few terpenes. There's been well over 200 that have been identified in cannabis. So this is one of the reasons that so many of the different varieties of cannabis and hemp have such unique scent profiles. Metabolically, they're secondary metabolites produced through the MVA, MEP pathways. So these pathways build these compounds from repeating units of isoprene. So these units determine whether there are monoterpenes or diterpenes or sequestraterpenes. It's just merely a designation of the number of units of isoprene used in their metabolic pathway. And so you hear this term chemovar a lot. It's become fairly popular, and I'll talk about it in a little bit more detail shortly. But basically, the idea behind this is it's sort of the chemical fingerprint of the strain of hemp or cannabis. So it's why certain strains make you feel or smell a certain way. So it is an important part of the hemp experience. Now, why should we care about terpenes? They they play a very, very critical role in the consumer experience. On a very interesting note, a lot of the winners from Cannabis Cups have unique and robust terpene profiles. So a very good analogy that I heard about this was just like, it's like the difference between a $4 box of wine and a $200 bottle of wine. Those alcohol concentrations are almost going to be identical, but one of them provides a much different experience, right? So just like you wouldn't judge these wines by how much alcohol is in them, people tend to not judge cannabis based off of the THC, CBD. Like you'll see that, a lot of times there's this focus on high numbers, but really, truly the experience comes from the interaction between these analytes and the terpenes. So this term has been coined as the entourage effect. And it's a synergistic interaction between terpenes and cannabinoids in the human body. So there's been evidence that shows that terpenes also interact with the endocannabinoid system. So it's not too big of a leap of faith to recognize that they play a part in the overall experience. So there's thought that, these compounds magnify the therapeutic benefits. A lot of times you'll hear people discuss why, when one strain makes them feel a certain way and another one, may make them feel sleepy, although that is a purely qualitative speculation, right? So it most likely has to do with the interaction of these compounds and their brain chemistry. So the idea is that the sum of each one of these parts is greater than the whole experience. All right, now before we get into analysis, we're going to talk a little bit about the sample preparation. One of the biggest struggles with analyzing hemp and cannabis is the broad variety of matrices that show up to our lab. So, CBD from hemp is put into everything imaginable, from shampoo to bath bombs, dog treats, toothpicks, beef jerky. We've seen everything you can imagine come through our doors. So this creates some problems. There's a term in analytical chemistry called a matrix effect. So this is sort of everything but the target analyte that you're looking for, which in this case are terpenes, and their interactions with your ability to measure them. So sometimes this can amplify, give you a false increase in values, or it can diminish the response from the instrument and you get artificially lower values. So, sometimes it takes us a little bit longer to get right on point with some of these analyses. So the sample preparation, we use 500 milligrams of sample. So in analytical chemistry, the larger the sample size that you have, the more accurate your results tend to be. This value comes from the Bureau of Cannabis Control regulations. So they require for compliance testing, you use 500 milligrams of sample. Now for hemp, there is not federally mandated regulations yet, but we've seen a significant increase in what we call a full panel test for CBD and hemp products. So these are all the heavy metals and terpenes, residual solvents, cannabinoids. These are all the required tests for cannabis, but we've seen people becoming more and more interested in this for their hemp products. And the idea is that they are delivering a safe product to the consumer, which is really our end goal as well. So for terpenes, for hemp flower extract, we typically use methanol. So we grind up these 500 milligrams, we dilute it to a total volume of 10 milliliters. This creates a 20-to-1 dilution. Sometimes we can run into some problems with oils, so we use a combination of DMA and methanol, and that seems to increase our extraction efficiency. So once the sample is ground and extracted, so usually we use like a mill bead, or if it's a gummy bear, we will freeze it in liquid nitrogen and grind it into a fine dust. And so once this is ground sufficiently, we add our extraction solvent. And then we take a 20 microliter aliquot out of that. And we use what's called total evaporative analysis. So the entire 20 microliters is evaporated into the gas phase. And so once this occurs, we're now ready for our analysis. Now I'm going to move on to the instrumentation now. This is our setup. So this is a PerkinElmer instrument. We, all the way to our right is the Headspace sampling system. So if you see on the top of the GC, there's also a liquid injector in this image. So I'll get into that a little more in a second. So this second box in the middle is called a gas chromatograph. So this is where our separation takes place. So chromatography is the science of separation. You've probably heard of a liquid chromatograph. This is a gas chromatograph. So both of those technologies are used to separate out analytes. And then the detector that we use in our analysis is called a mass spectrometer. So this allows us to get a mass spectrum of the target analytes, and it allows us to easily identify the compounds that we're looking for. So I'll get into that in a little more detail in a minute. So we're going to start with the sample injection system. So you can see all the way on the right, this is a 40-unit carousel. We have 110-unit carousel in our lab, which basically allows us to run continuously. So the sample is put into a 22 milliliter vial and sealed, and then it's rotated into the autosampler oven, where it's baked for 10 minutes at 160 degrees Celsius. So this allows us to inject only the compounds that we want to. So the majority of terpenes, so at least the ones that we look at, boil below 160 degrees. So everything that in the sample that boils above that will remain in the vial. So once all of these analytes are volatilized, we inject a specific volume into the inlet that is moves into the column. And so the alternative to Headspace is liquid injection, which is a much more messy methodology. So basically you can see on the right of this picture is a sample vial. There's a lot of chlorophyll and other compounds. So there's well over 1,000 compounds found in the cannabis plant. So not all of those we want to look at. You can see in that injection liner there, that's after only 7 injections. You can see all that sort of nastiness caught in the liner. So these need to be changed quite often, at least weekly. I've heard from certain labs, they change them every 25 to 30 samples. So this kind of creates a production nightmare. So Headspace is a much cleaner technology. I've heard of labs not cleaning their source or doing any sort of maintenance in this regard for multiple years with the total evaporative analysis Headspace technique. Now, you can theoretically measure samples directly with Headspace, which is pretty cool. So we've found that this works really well for the actual flower. So you can take the flower and put it in a vial and heat it, and everything that we want to measure will volatilize directly off of that flower. Now, this doesn't work as well for oils and some of these other complex matrices that we see. So once we have our sample volatilized, it's now injected into the gas chromatograph. So gas chromatography is an analytical technique that is used to separate out volatile compounds. So inside these columns, which are typically 30, 15, or 45 meters in length, they're wound very tightly inside here. And they have what's called a stationary phase, which is a thin film on the inside of these capillary columns, which our target analytes will interact with. So in chemistry, we always say like dissolves like. So if you think about oil and water, this kind of illustrates the potential interactions with these columns. So once we've injected, we have a program temperature wrap, which slowly elutes the compounds off of the column, one by one, into our detector. So in this next slide on the right is a typical chromatograph. So here, in this one, we're looking at 42 different terpenes. So there's well over 200 terpenes that have been identified in hemp. We have two different methodologies that we run in our lab. So for compliance testing and the majority of our analytical testing, we look at only 21 of these terpenes. From our experimentation, we found there's roughly 17 that are present in high concentrations in most hemp samples. So in order for us to run these 200 terpenes, it would take well over an hour, and on a $500,000 plus instrument. So it's not really feasible to measure all those compounds in a commercial type setting. So the runtime for 21 terpenes is about 10 minutes. We're a little bit hindered by the Headspace, so it's about a total from sample injection to complete data collection, it's about 15 minutes. So we can run about 70 plus samples in a day. And then 42 terpenes is a little longer at about 14 minutes. So once these samples, they come off one by one, we're able to collect spectral data using our mass spectrometer. So mass spectrometry is a technique that allows us to measure the charge ratios, or mass to charge, of individual ions. So once these are separated and they move into the mass spectrometer, they come in contact with what's called a source. So in this case, it's electron impact, which creates these ions. So it basically splits them into pieces, and then we use the instrument that we use for this is a single quadrupole. So it's 4 rods that utilize a oscillating electric field that only allow certain masses through. So it helps us eliminate significant noise from entering the detector, which allows us to have a quick runtime and then also be able to definitively determine what analytes we're looking at. So the detector in this case is an electron multiplier tube. So it basically amplifies the signal. And then we're able to collect a mass spectrum. So on the right here, you can see on the top and bottom, these are 2 typical mass spectrums from different terpenes. So these mass spectrums are basically like a chemical fingerprint. So we can run this against a database library and determine exactly which terpenes are in our sample. So even if we're not able to quantitatively provide a value, we can also, if we get other hits that we're not looking for, we can still determine what those terpenes are. And so our calibration, so terpenes typically are linear across a broad range. I think in this, we typically run from about 2 parts per million up to 500. Terpenes, they rarely exceed 1 to 2% by total weight in flower. Although with extracts, things like live resins, this sometimes can be an issue. So usually we use this 20-to-1 dilution, and it's sufficient for about, I would say 90+% of our samples. The highest terpene concentration I've seen in flower was 4%. So it does happen occasionally. And in that case, we just dilute and run it again. Okay. If you guys have any questions or comments or thoughts or want to learn more about any of this, I love talking about terpenes and analytical chemistry. So please feel free to reach out to me. My email is adam.floyd@think20labs.com. Thank you very much for attending my seminar.